In this presentation, we're going to look at, in particular, some of the amino acid neurotransmitters. And so just to refresh our memories what some of those are, here we have a short list right here. We can see GABA, glutamate, glycine, and histamine all fall under this category of amino acid neurotransmitters. But some of you asked a very good question, and that is, well, what's the difference between that and peptides? So I thought I'd just start with a little bit of a refresher on some of the biology of this. So we're going to look at our biochemistry. So let's go ahead and take a look. So here, as a refresher, remember that amino acids are the building blocks of all proteins. And proteins make up pretty much everything there in our bodies. Just a ton of things are made of proteins. And just the basic structure of any amino acid would be this, where you'd see this functional group, this called the amino or amine group here on the left, and then you'd see another functional group on the right. Remember, those functional groups tell us something about molecules here, and then we have a carbon in the middle, and then we have this thing called R. And R, if you remember, is the side chain for that amino acid, which means that this can represent all sorts of different things. And down below, you can't see these too well, I'm sorry, but here are the R groups of, right in here actually. Here's an R group, and another R group, and another R group for the three of the amino acids, phenylalanine and serine and proline. So everything else is exactly the same on these except the combination of things which represents that R group or the side chain. And so that's what distinguishes one type of amino acid from another. And there are about 20. There's actually more than that, but 20 that we really focus on in biology when we're looking at proteins. Now, so there are certain amino acids which are basically neurotransmitters, and we'll talk about those. But then there are also these things we call peptide neurotransmitters, or in some cases these can be also structures that are related to hormones. And when we talk about peptides, what we're saying is that a peptide is bigger than the amino acid types of neurotransmitters. And so peptides are a series of very specific amino acids that are bonded together by these bonds, which we remember are peptide bonds. So I picked two of them that I think would be, that we're going to talk about soon anyway, and that's this arginine vasopressin, and this is a presentation I believe that Jade gave us, and then this one on oxytocin, which is the one that Nikki gave us. So we're going to look at these coming up, so just stay tuned. So our real focus here is Michaela's presentation and Amelia's, and I think I'm going to go forward here, but we'll talk about each one in turn. So Michaela presented glutamate, and I talked about glutamate just the other day because glutamate is probably the most widespread neurotransmitter. Certainly it is the most excitatory neurotransmitter in the nervous system. And so what that really means is that when glutamate is released by a presynaptic neuron, the postsynaptic neuron, if it gets an ample activity on its side, is going to have an action potential. Now there's another one that's very similar to glutamate, and that's aspartate. And that might actually, if you know anything about some of the artificial sweeteners, you may have heard of aspartame as being like NutraSweet is one of those types of artificial sweeteners. So I'll talk a little bit about that later on. But basically these two, particularly glutamate, are excitatory. And when we say that there's these neurons are releasing it, and we talk about these neurons, we can call them glutamatergic. So when they release glutamate, the glutamate moves across the synapse, and the first thing it's going to activate are these AMPA receptors. And if you remember what we talked about the other day, AMPA receptors are found on these postsynaptic neurons, on the dendritic spines, or those little swellings and extensions on those dendrites. And AMPA receptors are ionotropic, so they're going to open very rapidly. They're going to let in ions. In particular, they'll let in sodium. So that's what glutamate does. Aspartate does basically the same. But we also know that glutamate can affect other types of receptors. And on that postsynaptic side, in many places, we have, in addition to the ionotropic type of receptors, those AMPAs, we also have metabotropic receptors. And those are called, well, they can be called a number of things. We'll talk about that NMDA receptor, which is one of these types later. But this M-GLU-R basically is saying that there are different types of metabotropic glutamate receptors. And they're slower because they aren't directly associated with ion channels, but instead they're going to cause a cascade of events to happen inside the neuron that leads to other effects. So we talked a little bit about this already, how abundant glutamate is and how important it is to understand in the nervous system because of its effects. One of the things that we've alluded to a few times is that glutamate, when it's released, and actually all neurotransmitters, particularly these classical ones we're talking about, they have mechanisms to quickly clean up the synaptic area, so to get rid of the glutamate that's there, to stop its activity. And we've mentioned some of the ways that happens. So it can be brought back into the neuron, the presynaptic one, by reuptake. But I wanted to just talk a little bit about the role of astrocytes. So remember, there are all these glial cells in the nervous system. We talked about oligodendrocytes, which are those ones that wrap around the axon to form the myelin sheath. We've talked about microglia, which are the ones that kind of clean up the debris, the sort of the little Pac-Man of the nervous system. We've talked about astrocytes as being associated with the blood-brain barrier and connecting neurons to the blood supply via these extensions they have, these little things called end feet, which we aren't seeing here. But here's another role of astrocytes, and a very important one, in that astrocytes also can pick up the leftovers. They bring in some of that glutamate that's in the synapse, and they do a whole chemical conversion of that glutamate back to something called glutamine, which is really what gets converted into glutamate in the first place. And interestingly, this also gets converted into GABA. So opposite effects. So we have these two, that from this precursor molecule, this glutamine, it can go back into a glutamatergic neuron and get converted back into glutamate and then be put in vesicles. But when glutamine gets shuttled back to a GABAergic neuron, it will also be made into glutamate as an intermediate product. And then another enzyme changes its structure and makes it GABA. So just a really interesting way that these astrocytes have this role in removing glutamate from the synapse, but also providing that once it's broken into the glutamine, the bringing back that compound that can then be transferred either direction, ultimately to make glutamate again, if it's a glutamatergic neuron, or can be converted into GABA. So back to glutamate. Glutamate, really important role. It plays lots and lots of roles in various places in the body. But we talked about this, at least initially, this role in memory, learning and memory. So it plays a very key role in these cognitive processes. If we have too much glutamate, though, if something causes glutamate to be released, for example, in a great amount, kind of diving down to the third bullet here, then that can result in excitotoxicity. So glutamate can overexcite neurons and therefore can cause the neuron to die. So this is part of why this mechanism we just talked about, getting glutamate out of the synapse and converting it, is so important. Some of the possible times this could actually happen, though, is when there's activity where the brain doesn't get enough oxygen. That can actually cause a release of glutamate from these glutamatergic neurons, kind of like something's happening here. During a stroke, that can be caused. During epilepsy, that can happen, and that can be toxic to neurons. If there's not enough glutamate, that means that it can't do some of the things it normally would do, at least not as effectively. There's association of imbalances of glutamate that are in the nervous system where we aren't making enough, with Alzheimer's disease and some other types of cognitive impairments. Now, I mentioned this once before. I mentioned this interesting thing about glutamate. This is that we actually take in glutamate, or at least we can, through a compound called monosodium glutamate, or MSG. Now, for most of us, our blood-brain barrier is pretty intact, and therefore what we take into our stomach and our diet doesn't necessarily go outside of our the area of our body and doesn't necessarily go into our brain. But in babies, that blood-brain barrier isn't particularly well formed, and so MSG could have a detrimental effect and cause some excitotoxicity if there was MSG in baby food, which I believe there is not anymore, but I think there was at one point some suggestion that that was true. I'd have to look that up in the history books. So now let's go to GABA and glycine. And so Nehemiah and Amelia both presented these neurotransmitters, both of them very powerful inhibitory neurotransmitters. And they both work pretty much in the same way, but they work in different places. So glycine is primarily in the brainstem and spinal cord, and GABA is very prominent in the central nervous system. Both of them activate chloride channels, and they inhibit neurons by bringing in the chloride. And if you remember that's going to cause, instead of depolarization, it's going to cause hyperpolarization. So it's going to go below that -70 mark and make it harder for a neuron to fire. As a matter of fact, it would, if that was all we were talking about, if it's only GABA or only glycine, it basically will inactivate that neuron so it cannot fire. But we'll talk about some of the other things here shortly. So GABA has a number of receptors. A lot of them are ionotropic because it's going to cause this really fast activity. But there is also a metabotropic one, which is the slower inhibitory type, but involving some type of G protein or internal mechanisms that cause other things to happen. Now, things like, there are some agonists to GABA receptors. One of them you may have heard of before is Valium. And Valium is something that will go in to a GABA receptor, and it will activate it, and it will basically slow that receptor, cause that hyperpolarization. So they are found in tranquilizers, things which we, to lower our activity rate in certain areas of the brain or body. So that would be Valium. There are also glycine agonists. And there's a whole laundry list of things that we could talk about here. And I put a little bit in the document I sent to you, but glycine is going to be activated by strychnine, which is a poison, and that can be a very fatal activity when those agonists are present in that part of the nervous system. So, and I already talked a little bit about how GABA and glutamate shuttle works. So I think we are at the end of this particular presentation.